This recorded lecture is dedicated to our discussion on cellular respiration. As we have completed already our introductory lectures on the origin of life, the study of zoology, and um, animal cell structure and function, it's now time for us to discuss metabolic processes happening within animal cells. So, cellular respiration is a metabolic pathway that produces energy in the form of ATP, primarily starting from glucose molecules or making use of glucose molecules as um, starter material. Okay, in animal cells, the process um, yields or is required for ATP molecules to be produced. Now, we'll look into several aspects first before we discuss the um, major pathways or major metabolic mechanisms that are known happening in animal cells for the ATP for energy to be produced. So let's first compare and contrast cellular respiration with photosynthesis. In photosynthesis, carbon dioxide in the presence of water is used to produce glucose and oxygen. So um, in the presence of the green pigment chlorophyll from that is found in all plants that captures sunlight, okay, carbon dioxide and water are reduced and oxidized respectively as glucose and oxygen molecules. So remember the difference, or remember the what happens for each molecule. So when we say reduction, um, we can appreciate, or when we say reduction, the process involves an atom or compound gaining one or more electrons. So that's reduction. So carbon dioxide is reduced to glucose, um, while water undergoing oxidation, which is the opposite of reduction, where electrons are lost. Okay, electrons are donated from the donor molecule to a recipient molecule. Okay. So I mentioned this already that there should be two important requirements present: solar energy and chlorophyll. No? Okay. Now, um, for the case of cellular respiration, you can think of it as sequential, or what happens after glucose is produced. But of course, in different systems, in animal cells. So glucose in the presence of oxygen, okay, can be broken down into smaller molecules alongside with the production of ATP. No? So glucose is oxidized such that it becomes carbon dioxide, while oxygen is reduced, so water is actually produced or manufactured. And in the process, ATP is produced. No? Okay. Now let's take a look at ATP as the central molecule of concern for in the context of cellular respiration. So here you can see the structure of ATP, where the part that contains the sugar, the ribose sugar, and then the base, the nucleic acid base. So this component here is what we call the nucleoside component. No? Attached to one phosphate group, this nucleoside component becomes or is known as adenosine monophosphate because there's only one phosphate group attached. If there are two phosphate groups attached, this becomes adenosine diphosphate. And ultimately, in relation to our discussion on cellular respiration, if a third phosphate group is attached to the nucleoside, that becomes adenosine triphosphate. So please remember the difference. It's very basic, but important for us to understand. Very important for us to remember that from AMP to ATP, the primary or the key difference is the number of phosphate groups that are attached to our nucleoside, adenine nucleoside molecule. Okay? Now, let's discuss the process of glycolysis. So for the remainder or for the entire of the lecture, we'll talk about glycolysis. Um, and for Saturday, I'll be discussing the electron transport chain. No, so glycolysis is a preparatory step for electron transport chain. In terms of the location or where it occurs, it takes place in the cytoplasm of the animal cell. Now, simply when you dissect the word glycolysis, so glyco means sugar or glucose, while lysis means breakdown. So simply put it, glycolysis is the breakdown of sugar to produce starter molecules for electron transfer chain for oxidative phosphorylation. And at the same time, energy is actually produced. No? So glucose is a 6-carbon molecule, and during the process of glycolysis, it's converted to two molecules of pyruvates, which are 3-carbon compounds. No? In most cell types, that's what is expected, but in others, ethanol can be considered, or ethanol can be a molecule that is involved during glycolysis. So as I mentioned, um, the breakdown of uh, carbohydrate is associated with energy production. The pyruvates that are produced during the process can be used in the succeeding cellular or in other cellular respiratory pathways, such as the citric acid cycle. No? Glycolysis is also known or synonymous with other terms such as the Emden-Mayer pathway. And as I've mentioned, it is a preparatory step for cellular respiration. Okay, let's take a look at to illustrate some of the key concepts that I have discussed so far. So as I mentioned, glycolysis happens in the cytoplasm of the cell. Okay, so here you can see the part of the figure or the illustration that is emphasized with the blue rectangle. Okay, so glycolysis, um, in summary, is breakdown of glucose to pyruvate, and alongside with the production of energy, ATP, and NADH, no, which is an electron, which is a compound important for the electron transport chain. In the presence of oxygen, okay, in the presence of oxygen, glycolysis serves as a preparatory reaction for the higher energy yielding processes that are occurring inside the mitochondria. So the pyruvates are transported to inside to the mitochondria along with NADH, such that these compounds will be used for higher energy yielding processes, such as the ETC. No? Then if oxygen is not present, all right, if oxygen is not present, pyruvate cannot be localized to the mitochondria, and instead it stays in the cytoplasm, and the net production of 2 ATP is expected, no? But essentially the yield is very low. So in the presence of oxygen, energy yield becomes higher, while in its absence, energy yield is actually limited. Okay? Very important to mention. So I mentioned already oxidative phosphorylation, uh, pyruvates being transported to the mitochondria. In the absence of oxygen, so pyruvate can be transformed to or can be useful for the lactic acid cycle. No? And energy is still produced, but then again, it's very limited. The amount is actually less compared, substantially less compared to oxidative phosphorylation. Okay? Now let's take a look at the processes or the events that are involved during glycolysis. So let's have an overview. Glycolysis is divided into two stages: an energy investment stage and an energy payoff stage. So from steps 1 to 5, that is the energy investment phase, while the succeeding steps until pyruvate is produced is the energy payoff or the energy release stage or energy production stage. Okay, so let's have an overview of the involved compounds, their transformations, and the involved enzymes. No? So, initially, glucose is phosphorylated to glucose 6 phosphate. So, that phosphate group is coming from an ATP molecule. The process is mediated or catalyzed by by glucokinases or hexokinases. So kinases are enzymes that facilitate the transfer of phosphate molecules from a donor to a recipient. So phosphorylation of glucose allows for the formation of a phosphorylated glucose intermediate, glucose 6 phosphate. Okay? The next step is for glucose 6 phosphate to transition or to form an isomer or to transform to an isomer uh, catalyzed by the enzyme phosphogluconase isomerase. Okay, so glucose 6 phosphate is transformed to its isomer fructose 6 phosphate. Okay, then the next step, step number 3, is phosphorylation of that fructose 6 phosphate, a secondary phosphorylation in a way, no? Because it will now contain two phosphate groups. In the catalyzed by the enzyme phosphofructose kinase, fructose 6 phosphate is transformed to fructose 1,6 bisphosphate, acquiring one phosphate group from another ATP molecule. Okay? Now, the next step before the energy payoff stage is the breakdown of fructose 1,6 bisphosphate into two molecules. These two molecules are glyceraldehyde 3-phosphate, PGAL, and dihydroxyacetone phosphate. So, fructose 1,6 bisphosphate is broken down into two molecules. PGAL, also known as phosphate glyceraldehyde or glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate. Now, if you notice, there is a double N arrow in between them, indicating that these molecules are actually interconvertible. And most of the time, the more stable version or the more stable molecule is actually glyceraldehyde 3-phosphate. So with the action of after the action of fructose biphosphate aldolase breaking down the fructose 1,6 bisphosphate into two molecules, another enzyme comes into play, which is triose phosphate isomerase. So this enzyme ensures that dihydroxy acetone phosphate is converted to PGAL. So you will have multiple molecules of glyceraldehyde 3-phosphate at the end of the energy investment phase. Okay? Then we enter the energy payoff phase. or stage, where we start with the oxidation of glyceraldehyde 3-phosphate into 1,3-bisphosphoglyceric acid or glycerate, with the corresponding reduction of NAD plus into NADH and 2H ions. Okay, so two molecules of glyceraldehyde 3-phosphate are oxidized, okay, into a different form, 1,3-bisphosphoglycerate or bisphosphoglyceric acid. And during that transformation, okay, an inorganic phosphate group is associated with NAD plus in order to produce NADH and H. Okay. Then the next steps 7 to 10, involve the generation of ATP molecules, a total of 4 ATP molecules, and the formation of 2 pyruvate molecules, which are also 3 carbon molecules, similar with PGAL. So what happens? So 1,3-bisphosphoglycerate is now further is now uh, in a way dephosphorylated because what its phosphate groups are donated to an ADP, two ADP molecules. So remember, there are two molecules. That's why think of these steps 7 to 10 as happening in two molecules. Okay, so 1,3-bisphosphoglycerate is transformed to 3-phosphoglycerate, and in order to do that, okay, transfer of ATP molecule, of phosphate group from the starter molecule occurs. Hence, the production of two ATP molecules. Furthermore, further down the line, no, 3-phosphoglycerate is transformed to 2-phosphoglycerate in the with the activity or the catalysis of phosphoglycerate mutase. In the process, water is actually released. No? Then lastly, right, so we have 2-phosphoglycerate converted to phosphoenol pyruvate via the action of enolase, and ultimately, this phosphoenol pyruvate is transformed to pyruvate molecules after it is dephosphorylated via the action of a pyruvate kinase. So the remaining phosphate group in phosphoenol pyruvate is now donated to ADP molecules to produce ATP. So the net production in terms of ATP molecules is four ATP molecules. However, because we invested, or there is an investment of 2 ATP molecules during steps 1 to 5, the net ATP that is produced is only 2. Okay? So, we'll look into the molecular events. Okay? But before that, I think the recording is already more than 20 minutes. I'll pause here.